In order for a cell to make a protein, the cell must first go through a process of protein synthesis. Within protein synthesis, the first process is transcription. In order for transcription to be initiated, within the promoter region, a promoter chooses a template strand of DNA. Also within the promoter region, there is a top box which picks an RNA polymerase. As the RNA polymerase starts at the promoter region, it unwinds and also translates the template strand into a new strand of mRNA. As we know in DNA, the nucleotides are paired off A to T and G to C. Instead, in the RNA strand, A is now paired with U instead of T. As the RNA polymerase moves through the DNA strand, it recreates the double helix shape. The RNA polymerase transcribes the DNA until it reaches the polyadenylation signal. At this signal, the mRNA that will be translated into a protein is cut out and it produces a pre-mRNA. With the production of pre-mRNA, the RNA must go through a modification before it can leave the nucleus. The pre-mRNA will receive a 5' on the 5' end and a poly A tail on the 3' end. The 5' tells where the starting point of the mRNA is, and the poly A tail protects the mRNA as it will travel through the cytoplasm.
Before translation can be started, initiation factors will bring together the two subunits of a ribosome, tRNA and mRNA. The tRNA is made with the help of aminoacetyl tRNA synthetase. This enzyme matches up a specific amino acid with the anticodon on the tRNA. The tRNA will enter the ribosome from the A site, where it will match up with a specific codon on the mRNA. A codon is a 3-nucleotide base on the mRNA whose complementary bases are located on the tRNA, which is called the anticodon. GTP, also known as energy, is needed in order for the tRNA to move accurately and efficiently from the A site to the P site. At the P site, the amino acid on the tRNA will be translocated to the A site's tRNA that has entered the ribosome using peptidyl transferase. Also, the tRNA in the P site will be located to the E site so that it will be moved out on the ribosome. This process will continue until a stop codon reaches the A site of the ribosome. When the stop codon reaches the A site, a release factor is added to the A site. With the addition of the release factor, there is an addition of water to the bonding of the amino acids. Through the process of hydrolysis, this breaks the peptide bond between the current tRNA at the P site with the polypeptide chain. This will release the polypeptide chain from the ribosome as it travels through the large ribosomal tunnel. After translation is complete, the ribosome structure will break apart. This break also requires 2 GTPs. So the free polypeptide chain begins to coil, which is the primary structure. At the quaternary structure of the protein, the overall protein is made by the aggregation of polypeptide subunits.